Okay. So now what I'm saying to all of you is we will start discussing all the questions very slowly. I will explain each and every question to all of you. And I'll give the time for every single question 30 to 40 seconds. Make sure that you people will read the question very correctly. See, if I am not audible, that is your problem only. Raju, you just refresh once again and rejoin. Rejoin once again. Rejoin. Okay. Now, every one of you listen. I'll give the time for all of you. Just read the question very carefully. Then only keep on answering the questions. And listen to me, even though you don't answer even a single question also correctly, you don't need to worry about that. This is all a kind of a practice questions, which will help you in understanding the concept in a better way. And everyone listen, whatever the questions that we are going to discuss right now, if good evening all, if you can answer this kind of questions, trust me, your question paper of JL won't be beyond this particular level. So imagine or consider yourself, this is a kind of a test that we are going to write in the JL. So make sure that all the questions are very easy. Read it thoroughly, understand it thoroughly. Don't give me the spontaneous answers. Always analyze the given question, then only give me the answers. Okay, now let's start the question number one. Question number 149. Every one of you start now. Just type the answers. That's all. Don't do anything else. I will explain everything. The question says that dark grown seedlings display the triple response when exposed to ethylene. That's the question. Dark grown seedlings. Slowly, I'm telling you just now, Maheshwari, don't be hurry. Listen my statements, don't be hurry. See, you people has to control your anxiety levels in the examination, especially in the competitive. Be patient, even though you are intelligent, even though you know the answer, just put your entire efforts in reading the question. That is what I'm expecting. I don't want your right answer or wrong answer. Be patient and analyze the question properly. Dark grown seedling display the triple response when exposed to ethylene. And the question says that which one of the following is not a part of the triple response? That's the question. Yeah, think and answer. Even though you know the answer, read the question again and then you give the answer. Yeah, type answers, type answers. Yay. Oh, really? Others, everyone. Right or wrong doesn't matter. I want every one of you give me the answers. B. Okay. What about others? Everyone type the answer. Every one of you. Right or wrong doesn't matter. Everyone give the answer. So perfect. So see, I hope you all people remember, right, friends, which hormone is actually involved in the process of the triple response. We all people know that the hormone ethylene is the one which is involved in the triple response. I told to all of you very clearly, decreasing in the epicortal elongation or inhibition of the epicortal elongation. That is one of the major function followed by that particular ethylene. So this statement is a right statement. The question they ask is which is not a part of the triple response. Do not get confused with this particular point, friends. Slowly think it. Thickening of the shoot, of course, that can be swelling. Thickening means here swelling. The swelling of the shoot also caused by what? Raju, it's the problem from your side. I'm telling you very clearly. Rejoin. I'm telling you multiple times, right? Please do not interrupt me in the middle of the class. Make sure that your connection will be stable. Okay. Thickening of the shoot, it is obviously performed by the ethylene. Horizontal growth of the epicortal. You know, what do you mean by the word horizontal growth here? Sometimes, the growth will be in this pattern. Even that can also be shown by the ethylene hormone. See, originally all the epicortal will grow like this in a straight manner, vertical manner. Sometimes because of the ethylene, what's going to happen? There will be horizontal growth occur. So that horizontal growth of the epicortal can also be caused by the ethylene. There's no doubt about it. And the point is very clear. The hormone ethylene do not have any direct or indirect role with respect to the expansion of the leaves. with respect to the expansion of the leaves. So the correct answer will be B. Don't get confused with that particular point. Okay, so now next question. Question number 151. Think it. You take your time, guess it. Don't do the random guesses. Give me a valid guess. Someone is making the sound. Who is that? Raju, it's yourself only. Don't make any noise. I will remove it. I'll remove you immediately if I get any other sound. Don't disturb me in the middle of the class. Okay, good. So yeah, what about others? Everyone give me the answers. So the question says that the okay, others. Very good, very good. Perfect. So you people consider that this is only your JL exam. Based on that, I want you to try to give the answers. The quantum yield of oxygen evolution during photosynthesis drastically drops in far red light. This effect is known as. First of all, I want every one of you understand what exactly the word quantum yield means. I want every student has to understand how can I define the word quantum yield, how many number of oxygen molecules are produced. Don't get confused. How many number of oxygen molecules are produced per photon absorbed. For a single photon that is absorbed, how many number of oxygen molecules are producing, that is called as the quantum yield. So I want you people to understand quantum yield will be constant up to which nanometer? The quantum yield will be absolutely constant up to 680 nanometers. So, once the quantum yield increased above 680 nanometers, it starts drastically drops down. It starts declining very sharply. So the point, the area where the decline is happening, that is only where, what do we call it as? We call it as a red drop effect. Once again, remember the point at which the quantum yield declines sharply, that point only we can call the red drop effect. Wonderful. So don't be hurry. Think and answer the question. Question number 152. The question says that nitrogen gas is reduced to ammonia. Slowly. Is reduced to ammonia by a nitrogen fixation method in order to execute the process. Which one of the following is usually required? So it's a simple, straightforward and direct question. We all people know very well that what is the major enzyme involved in this particular process of conversion of the N2 to NH3? You know that we are converting N2 to NH3 or NH4 plus. That's what we generally call it as the nitrogen fixation. And what is the versatile enzyme involved here? The versatile enzyme involved here is the nitrogenase enzyme. And the point is very clear. The nitrogenase enzyme, we discussed very clearly. Nitrogen fixation is a highly energy consuming process, which means that it's gonna use abundant amount of the ATP. Now, you all people has to ask me a question. Sir, actually GTP, UDP, ADP also energy precursors, right? Then why it can't use? That's a very good question. The question is, GTP also provide the energy, ADP also provide the energy, UDP also provide the energy. But the point is that why nitrogenase enzyme use only ATP? That's a very good question. Because friends, nitrogenase enzyme having a binding site only to the ATP molecule. Only ATP can associated with the nitrogenase enzyme. No other energy precursor cannot associate. That's a simple answer. So the correct answer will be the ATP. Don't be hurry. I want you to think it. Easy questions anyone can answer. I want you to think and give me the answers correctly. Question number 153. Just a minute. Yeah. See, think it. Take your time. Symbiotic nitrogen fixation in legume nodules involves complex interaction between rhizobium and legume roots. This complex interaction is governed by. It's a very good question. I want you to think it. And they're asking the question which is correct, my friends. They're asking which is correct. Easily you can analyze. It's not at all difficult. Slowly. See, everyone is giving fast answers. Please don't give fast answers. Improve your patience. Don't give me fast answers. Yes. You can see it now, no, Suma. If I zoom it, you can't see the option. Okay, now it's fine. Take your time. What will be the answer? Most of the people are saying B is the answer. Okay, think it, think it, think it, think it, think it. Slowly, slowly analyze. Don't be hurry. Okay, what about others? Everyone give me the answers, whether right or wrong, doesn't matter. Give me the answer enough. Anuradha, it's A. How is that possible? Okay, all of you look into the given question. They're asking the question, how does the bacteria and leguminous roots are going to get associated? I have told to all of you multiple times, right, friends? I told you very clearly that, listen everyone, the root hair of the plant, what the root hair of the plant is releasing, it is releasing certain chemicals called as a chemoattractants. We studied an example of chemoattractants, flavonoids and betaines. Now you tell me, if you look into the option A, says that integration of the symplasmid of rhizobium in the root nuclear genome. First of all, tell me what's the use of the plasmid here? What's the use of the plasmid here? We don't have any direct role or an indirect role of the plasmid in the nitrogen fixation process. Then there's no point of using the plasmid here. Obviously, option A is wrong option. If you analyze that option A is a wrong option, undoubtedly you can come to the conclusion that the option B, C, D are absolutely right. So what the plant is going to do? So sensing the plant flavonoids by bacteria. I told you na, flavonoids only chemoattractants. See, I am a root. When I release certain chemicals into the soil, the bacteria will attract. Then bacterial body, what kind of genes are going to activated? Nod genes are going to get activated. I have very clearly explained it to you. The genes which are present in the bacteria are called as nod genes, and the genes which are present in the plant we can call it as nodulin genes. But the point is crystal clear. If the nitrogen fixation wants to take place, it is compulsory, it is mandatory that there is a requirement of nod gene also, there is a requirement of the nodulin genes compulsory. Hence option B is correct, C is correct, D is also correct. Hence what will be the answer? The answer will be the C. There's no role of the plasmid in the nitrogen fixation. Don't get confused with this particular point. Okay. Okay. See, friends, you people take your time. I want you to read the question correctly. You people can answer it easily. It's not difficult question at all. Think it, use your brain and answer. 154. Don't give me fast answers. You can answer it easily. Still people. Okay. I have, this is the maximum zoom we can do. It's not at all difficult question. It's very easy. Take your time. No problem. Don't give me the fast answers. Naj, please don't give me the fast answers. You know what my worry is? You people are not reading the question even 30 seconds. I don't want your correct answers. Even you give all questions wrong answers, no problem. You read the question, analyze it correctly. Don't be excited. Always remember, reading is different, revision is different, practice is different. Question solving is a skill. It's an intelligent skill linked with patience. Always be patient, keep your mind calm and answer. All the questions are not same. You know it very well. At least one fellow has to give me the answer. Okay. Every single line is there in the PPT. Okay, Seema said D. D. Okay. Okay, look into the given question, every one of you. First of all, everyone answer my question. Everyone answer my question. What is red light photoreceptors here? Every one of you answer. What is the red light photoreceptor? Undoubtedly, friends, the red light photoreceptor nothing but phytochrome. Yes or no? And we all people very clearly studied that phytochrome is coded by a gene family. Yes or no? You all people studied A, B, C, D, E. Yes or no, friends? I have very clearly explained to you A, B, C, D, E genes. So option A is absolutely true option. There's no doubt about it. If you comes to know option A is the correct option, you can eliminate option B. You can eliminate the option C also. Now you look into the option 2. Option 2 says that phytochrome C is the most prominent photoreceptor to perceive red light. No. Among all the phytochromes, which is the most important phytochrome, phytochrome A is the most important one, not the C. Hence, the option B is wrong answer. Then only you can come to a conclusion, answer D. Now, I have very clearly told you, friends, cryptochrome contains what? Cryptochrome contains apoprotein and it contains chromophore. What is this apoprotein? I have explained you 5 minutes about this only. The apoprotein, they look like a bacterial DNA photolyase enzyme, but they do not have any kind of a DNA repair activity. So C is right. And far red light is not perceived by the phytochrome because phytochromes are only absorbing the red light. Such a simple question it is. So what will be the answer? Answer A is correct. Answer C is also correct. Hence the correct option will be D. No confusions. So slowly analyze the question. Be calm, be patient, slowly. I don't want your right answers. Just read the questions, enough for me. Question number 163. Oh, yeah, if I zoom it, you can't see it. Okay, this is the question number 163. If I zoom it, you can't see it. Following are certain statements regarding terpene class of secondary metabolites in plants. Don't confuse, think and answer, okay? Isopentenyl diphosphate and its isomer combine to form large terpenes. Diterpenes are 20 carbons. All terpenes are derived from the union of 4 carbons. Option D says that pyrethroids are the monoterpenes. You people has to give me the combination of above statements which are correct. Slowly analyze, slowly. If you know one option also, you can give the answer for this question, even though you know one answer. Seema is very fast. Okay, what about others? Okay, everyone has given the answer. That's great. So you know, right? Isopentenyl diphosphate, which is called IPP. Okay, I have told to all of you many number of times. IPP and its isomers. See, correct? No, abundant amount of IPP molecules come close together and it is going to give rise to a larger terpenes. Absolutely true point. There's no doubt about this. Right statement. Diterpenes are what? Diterpenes are 20 carbon containing compound. I hope you all people remember. What's the example for the diterpene, friends? GGPP. Ziranyl diphosphate is the example, which is a 20 carbon. Absolutely right. A is right, B is right. Now, if you look into this, all terpenes are derived from the union of 4 carbon. No, right? All terpenes are derived from the 5 carbon, which is called isoprene unit. So C is wrong. Pyrethroids are monoterpene. That is also true. Hence, the correct answer will be B. Wonderful, wonderful, wonderful. Okay. This question is little tough. It's not tough. Whatever we studied, the same thing. But you people, if you remember the concept correctly, you can answer. If you don't remember, you cannot. Question number 165. You can answer. That is not a very difficult question. I don't know whether you remember the signaling or not. If you remember the signaling, you can answer it. And I told you also very clearly. This is the diagram which is there in the slide. From the slide only I have explained to you. If you can answer this kind of questions, trust me, you can answer any kind of a question that is coming in your JL. Good. Be calm and analyze it. Eliminate one option. It's okay. Try it. This much fast, Ramya, you read all the statements. I haven't read all the statements only. Okay, good. You know, friends, once we keep the option in the exam, we can't take it back. Is it? So that's what you please be patient. Slowly analyze. Yeah, Mahipal Reddy, what will be the answer for this question? Yeah, Lakshmi said D. Okay, see, there are two things should happen here. If you really studied the ethylene signaling, this question is not at all difficult. If you don't know about ethylene signaling, I mean to say, if you did not study, you are doing a random guess. So trust my words, random guess are never going to work, because this question is not depending on your guessing. This question is depending on the concept. Now look into the question. The statements are given about the signaling of the pathways. I have told to all of you nearly 10 to 15 times. Friends, you should always remember what happens in the plant if there is a hormone. What happens in the plant if there is a no hormone? I told you that is what entirely signaling is depending on. Now you look into the option A. It was written that unbound ethylene receptor work as a positive regulators of the response pathway. You only try to understand ethylene is not there. Ethylene is not there. Then friends, how is that possible? The positive response can occur. Undoubtedly, option A is wrong option. If you understand option A is wrong, you can eliminate option A also. You can eliminate option D also, because you have to understand without hormone. See, I am a hormone. When I go and attach to the specific receptor in the plant cell, then only the response can occur. If there's a no hormone, how does the response occur? There is a no response. And I don't know whether you remember option B. I have very clearly told to all of you that there are more than 2 ethylene receptors are known to date. Actually, I told you, friends, there are more than 5 ethylene receptors are synthesized, and especially we found in the Oryza sativa and we found in the Arabidopsis thaliana. I have explained you the same in the slide. So option B is the right option. Option C, how many number of times I told you? Friends, all the hormones are following what kind of receptor? Multiple times I told you, they are following the histidine kinase domain. This is how the enzyme linked receptor is looking like. If anyone forgot, go to the signaling part once again, read it. So here all the histidines are present. So that's what I told you, histidine kinase domain is present here. Option C is also right. Option B is right. Option C is right. Then the correct answer will be the B. Only one fellow, I think Seema, has given the correct answer. Now, Revati, look into this. Why the option D is wrong? EIN2 is a transcription factor. EIN2 is a transcription factor. But what is the statement they have given in the question? You only see here. They are saying that if the transcription factor got mutated, then the response of ethylene occur. That statement is absolutely wrong statement, right? If a transcription factor got mutated, how does the response occur? Response never takes place. But they said that it promotes the response. Option D is also wrong. Hence, the correct answer will be the option B and option C. Hence, this will be the answer. That is B. No problem at all. Nothing is going to happen. So now I want you to read that once again after the class. I'll share this. Next, which one of the following plant hormone uses two-component histidine kinase receptor system for the signal transduction process? Easy question, and you know it. Don't be hurry. Haven't studied the question. People given the answer. So, which of the following plant hormones uses the two-component histidine kinase receptor system for the signal transduction? Undoubtedly, I told you, right? Cytokinin. I have explained you, friends, many times. Suppose example in the exam, cytokinin and ethylene given one option only. Then obviously choose this option only. But if the cytokinin and ethylene given in the two different options also, I want you people to choose the cytokinin only. But in the given four options, if the cytokinin was not given, then you people go for the ethylene. Don't get confused with that. It's very, very easy to understand. That's perfect. Okay. I want every one of you to answer this question. I don't agree if you don't answer. It's very easy. You all people studied. I have explained it to all of you 5-6 times. The following statements are made to describe auxin signal transduction pathway from receptor binding to the physiological response. You read it. Think it. Hope at least one fellow given the answer. Seema, very fast. You're analyzing correctly. Okay, others? It's not difficult question. It's very easy question which we studied. The point is that you people forgot. Yeah, Revati, what is the answer? Varalakshmi said, Naz said A. Okay, Nurin A. Okay. Others, Jyoti, what's the answer? Yeah. Mahip already said yeah. Okay. Now look into the given question. What's the question says that? We studied this very correctly. Auxin response factors. What do we call ARF? I told you very clearly in the class. Friends, without auxin response factor, there is no response of the auxin only, right? So auxin response factors are the nuclear proteins will go and bind to a particular sequence in the nucleus. What is that sequence called as? Auxin RE sequence, auxin response element. Absolutely right statement. There is no doubt about it. Now look into the point. Auxin IAA proteins are secondary regulators of auxin-induced gene. What did I told to all of you? Friends, auxin IAA, I told you, right? They are called as early genes or they are called as inhibitory genes. What is the function of a auxin IAA early genes or inhibitory genes, friends? They are actually going to block the ARF protein. Yes or no? Obviously, yes. Right. So now you look into the option. So option B is also right because auxin IA agent block the ARF. That is true point. Option C says that auxin bind to a receptor called TIR1. I already told you very clearly what we discussed. The moment auxin comes and attaches with the TIR1 receptor, what's going to happen? Auxin immediately activate the ubiquitin system. That ubiquitin system will go and associated with this targeted genes. Then immediately, friends, the targeted genes are going to undergo the process of degradation. I hope you remember by 26S proteasomal complex. Undoubtedly, option C is right. But what's the fourth statement says that auxin binding to the ARF cause the destruction by the 26S proteasomal complex. See, friends, the question is, they are saying that when auxin binds, ARF will be degraded. That is wrong statement, right? If the ARF degraded, how does the auxin response will occur? Never ever happens. So the point is crystal clear. Whenever the auxin binds, listen my statements very correctly. Whenever the auxin binds, always the early gene is going to get degraded, not the ARF. Without ARF, there's no response of auxin. Hence, option A is correct, B is correct, C is correct. Wonderful. Hence, the correct answer is A. Very, very good. Very good. I am not giving you the options also for this question. I want you to answer. Everyone type like this. Everyone type like this. Suppose A1, B2, C3, like that. Type entire answer in one option only. Type entire answer in one option. Take your time, no problem. Type everything in one option. A3, B4, C1, D2. Okay. Others, take your time. Take your time. Thirty seconds more. Everyone has to answer. Okay. We all people studied multiple times, right? The moment you heard the name of a auxin, the common point has to strike in your mind that auxin is the hormone which is involved in the polar transport. What is polar transport? I have very clearly told you, right? The unidirectional transport of auxin. That is what exactly we can call polar transport. Okay. So now listen my statements correctly. Then what will be the next one? Gibberellins. We already studied very clearly, friends. Gibberellin is the hormone which is involved in removing the seed dormancy. I hope every one of you remember, friends. What is the hormone that promote the seed dormancy? I hope every one of you remember. The hormone abscisic acid is the one which is actually promoting the seed dormancy. But they have asked for the removal of the seed dormancy. Obviously, the B will be the 4. Cytokinin. How many number of times I told to all of you? Cytokinin is the hormone which delayed the leaf senescence. Every one of you answer my question. What is the name of the hormone that enhances the leaf senescence? Everyone, what's the name of the hormone that enhances the leaf senescence? I hope you all people remember, right? Ethylene. Ethylene enhances the leaf senescence. Cytokinin delay the leaf senescence. And undoubtedly, here for the ethylene, what will be the answer? Epinastic bending of the leaf. So this is called bending of the leaf, right? Exactly. So what will be the answer? D will be the 2. Simple. So what are the correct answers? Quickly look into this. A will be the 3 and B will be the 4. C will be 1 and D will be 2. That's the correct answer. Perfect, perfect, perfect. Okay. Don't be hurry. Slowly think and answer the question. 186. Slowly. Answer is B. What's the question? It's very simple. If you analyze one option, you can answer the question very easily. Okay. What about others? Okay. Now, friends, please look into the given question. The given question says that the following are the statements that are made regarding secondary metabolites of plants. Option A says that all the secondary metabolites are constitutively produced in all the cells during entire lifetime. That is absolutely wrong. You think about our immune system in our body. In our body, do you think our body and every time produce the antibodies? That never happens. Is it or not? Whenever the antigen is entered in our body, then only our body start producing antibody. Similarly, in the plant, secondary metabolites are present, but they are not constitutively means continuously. They are not continuously produced or not continuously activated. Whenever there is a requirement, then only they are going to get activated. So option A is the wrong option. They serve as a signals to help the plant to survive in adverse condition. Of course, true. They may be volatile. We studied many examples. They contribute the flower color. You can take an example of anthocyanin. Obviously, the answer will be B. Very good. B, C, D. Fantastic. Question number 187. You know the answer slowly. Anyone remember? Very good. Very, very good. Very good. Hope at least one person also remember. Very good. Wonderful. So what will be that? Ethylene. We studied, right? When methionine is the precursor that is required for the ethylene biosynthesis. Is it or not, friends? Whenever the hormone methionine is getting converted into the ethylene in the intermediate, as an intermediate product, it's going to produce what? 1-amino-cyclopropene 1-carboxylic acid. Obviously, this will be the answer, will be the C. Very good. Wonderful. Wonderful. Don't be confused. I want you people to think correctly and answer. What's the question says that? The question says that following are the few statements regarding the structure of the terpenes. Take your time, slowly analyze and give me the answer. 1 second. Yeah. Option D will be C and D. Most of the people will make mistake here. I want you to think it correctly. I'm sorry. Sorry. Yeah. Think and answer. Think and answer. Take your time. Take your time. Don't give me the wrong answer, please. Mahip already said B. Okay. What about others? B. Everyone is saying B. Okay. Anyone else gonna give the answer? Varalakshmi said B. Okay, Jayalakshmi also B. Okay, now look into the given question. Look into the given question. The question says that isopentenyl diphosphate and pharancyl diphosphate are monoterpene and sesquiterpene. It is wrong, right? I don't know whether you're remembering or not. Isopentenyl diphosphate is how many carbon? 5 carbon only, right? What do we call this 5 carbon containing compound? 5 carbon, we generally call it as the hemiterpene. Monoterpene means how many carbon? 10 carbon, right? Monoterpene. But IPP is how many carbon? It is a 5 carbon only. Then how can it will be the correct answer? It is the wrong answer. Pharancyl diphosphate is a sesquiterpene 15 carbon. That is right. But one statement is wrong. Option A is wrong. Option B, squalene and zeroanyl diphosphate are triterpene and monoterpene. Even though you don't know the squalene, zeroanyl diphosphate, we studied, right? The GPP. Zeroanyl diphosphate is a 10 carbon. And I'm requesting all of you to remember that squalene will be the triterpene, which is having 30 carbons. Option B is right. Option C says that dimethyl diphosphate. How many carbon dimethyl diphosphate? It is a 5 carbon. And zeroanyl diphosphate 20 and 10. Zeroanyl is 20 carbon. There is no doubt about it. But the dimethyl diallyl phosphate is how many carbon? 5 carbon. But they said 10 carbon. Wrong statement. Diterpenes are 20 carbon. Sesquiterpenes are 15 carbon. Option B is correct. Option D is also correct. Hence, the correct answer will be the B. Wonderful. Good. Perfect. Perfect. Okay. Now the question says that the plant hormone gibberellic acid is generally not associated. They are asking not associated. Everyone given the answer also. Option D. Everyone forgot. Everyone forgot. Everyone forgot. See, friends, we already studied, right? Gibberellic acid is one of the major hormone that plays a crucial role in what? Stem elongation, of course. I told you that auxin is involved in the parthenocarpy and gibberellic acid is also involved in the parthenocarpy. There is no doubt about it. Of course, friends, in many countries, gibberellic acid plays a crucial role in the production of a malt. There's no doubt about it. Alcohol production. Which is, please mute your mics. Then parthenogenesis will be the answer, right? Gibberellic acid do not have any direct or the indirect role in the parthenogenesis. Very good, very good. Don't be hurry. Think and give the answer. Question number 196. Which one of the following statement is look into the question? Not correct for the Casparian strip. Not correct. They are asking. They are not asking which is correct. Easily you can analyze. Slowly, man, slowly. Yeah, Sri Vidya, what will be the answer for this? 196. They're asking which is not correct. Not. Okay. Look into the given question. Given question says that which one of the following statement is not correct for the Casparian strip? First of all, you all people know very well about the Casparian strip. What is Casparian strip? First of all, Casparian strip and where it is located. You all remember, right? Casparian strip is located on the endodermis. Yes or no? And we also studied that the Casparian strip contain what chemical? It contains the chemical called as suberin. What's the function of a suberin, friends? Suberin block the water molecules also. Suberin can block the lipid molecule also. Yes or no? Then obviously now you look into the point. See, whichever the water molecule that is following the symplastic pathway, there's a no worry because they won't face this Casparian strip at all. So option A will be what? Absolutely a true option. It breaks the continuity of water movement through the apoplast. That is true. Listen, everyone. And option B says that it is formed in the growing part of the root. Now tell me, I am telling you, right? It is located in the endodermis. Correct, na? How can you say that it is present in the growing part of the root? Do you think endodermis keep on growing? Never, right? Endodermis never ever continuously grow, friends. It's a fixed part. Yes or no? Hence the correct answer will be B. It is formed several millimeters or several centimeters behind the root tip. Yes, this is right. Now, of course, friends, here you're not understanding the question. What is suberin, friends? What is suberin? Suberin is a type of a lignin only, right? Suberin is a type of a lignin molecule only. Obviously, this is also a right statement. Hence, what will be the not correct is the option B. It never formed in the growing part of the root. Hence, the correct answer is B. Don't confuse. Don't confuse. Okay. Now, next question. Next question 198. The question says that given below are some statements on secondary metabolites in plants. Okay, slowly think it, analyze it, and give me the answer. Slowly. Take your time, no problem. Take your time, no problem. You have to choose the correct statements. Correct, you have to choose. Easy, easy it is. Take time, take time. Someone is giving the answer already. See, oh, really? Anuradha said D. Slowly. Seema, recheck the answer. Okay. Everyone, if you look into the given question, the question say, the option says that, listen, glucosinolates are synthesized by elongation in the length of the side chain of their precursor amino acid. I don't know whether you remember or not. What is the statement I told to all of you friends? All the nitrogen containing compounds, actually they are derived from what? All are exclusively derived from amino acid only, right? Of course, option A is right. No doubt about it. If you come to know that option A is right, you can eliminate option A, you can eliminate option C also. All terpenes produced in the herbs and spices are sticky, oily liquids which reduce the palatability. That's a very good question. A very good point. I have told you 4 to 5 times. What is the point that I have told you 4 to 5 times? Friends, secondary metabolites do not have any direct role or indirect role in the growth of a plant, in the photosynthesis of a plant, in the reproduction of a plant, in the life cycle of a plant. It do not have any role. But what is the statement says that? The statement says that all the terpenes are going to reduce the palatability. So how is that correct answer? This is the wrong statement. Option C says that cyanogenic glycosides are produced and stored in a toxic form. Now you only try to understand whatever the cyanogenic glycoside produced, if it is stored in the toxic form, first who will die? Plant will die first. I told to all of you, friends, all the secondary metabolites which are present in the plant body, they will always stored in the plant in a non-toxic form or in inactive form. Obviously wrong statement. B is wrong, C is wrong. Option D says that undoubtedly every single secondary metabolite can act as a defense molecule. It can be act against the herbivore also. It can act against the insect also. D is also right. Option A is correct. Option D is correct. Hence the correct answer will be D. Simple question, right? Okay. Is there any requirement of options here? 1 second, wait. I will only tell here. Option A, something is missing here. 1 second, 1 second. Yeah. Option 1, option 2, and option 3. You only tell me now. No need of options also. Just type the answer exactly. Easy, right? You can easily answer the question. It was wrong. It is xylem tracheary elements. Okay. The line got not properly typed. Okay, why others are not answering C? See, I want you to remember, friends, all the students listen my statements very correctly first. You know, right? Vacuole. See, this is the vacuole. How many times we discussed what is called the outer membrane of the vacuole? The outer membrane of the vacuole only we call it as the tonoplast. So every one of you remember my statements. Whatever the transport that is happening in the vacuole outer membrane, listen my statements. Whatever the transport that is taking place on the tonoplast of the vacuole, that is only we can call the transcellular transport. The moment you heard the name of a transcellular transport, remember that it's a vacuole transport. And apoplastic means symplastic, you know, right? Via plasmodesmata. Apoplastic means through the cell wall and intercellular spaces. So A will be 2, B will be 1, and C will be 3. Perfect. Simple question. So this is called as analytical question. Actually, this kind of questions you people never ever get in your JL. Okay, there is no doubt about it. You people never get it. But but it's okay. It's not required. Trust me, you people never get it. It's a very highly experimental based questions. We don't get it at all for the intermediate exam. Okay. Okay. Question number 218. They're asking, friends, which is not active. Okay, that's a good question. We studied now, friends, GA1 is, see, I have told you, friends, there are more than 150 type of the gibberellic acids are identified or isolated, but out of 150 gibberellic acid, friends, only the few type of gibberellic acids are extremely or biologically active. That is what we all people studied. So I want you to listen. GA1 is active, GA3 is active, GA4 is also active, but the GA5 is not active. So what it is going to act as? It is actually acting as a precursor. Correct? Now it can act as a precursor for the activated forms. Otherwise, it will be in the inactive form as well, right? Hence, the correct answer will be D. Don't get confused. Think it, slowly analyze, answer. Question number 219. Nitrogenase, a complex metal containing enzyme involved in the conversion of N2 to NH3. Which one of the following, see the question, which one of the following metal is not involved? The question says that which is not involved. Slowly, friends, slowly. Don't be hurried. No one will listen to me. Okay, don't be hurried. See, when I say that don't be hurried, the question is logical. You have to listen that. Okay, so Padmaja said that option D. See, that's what I want you to listen to me very carefully. See, you know, there is a bacterium called as Rhodopseudomonas. There is a bacterium called as Rhodopseudomonas. What's the speciality of this particular Rhodopseudomonas? The Rhodopseudomonas also containing the nitrogenase enzyme. There is no doubt about it. That nitrogenase enzyme, instead of using the molybdenum, it will use as vanadium. Right. So molybdenum can be used in the by the nitrogenase, iron can be used, vanadium can be used, which cannot be used is cobalt. So that's what, slowly, don't think that we can answer the questions very easily. Okay. Here you are not answering a question. The reason is that because you think that the vanadium cannot be used by anyone. That's a good point only. But there are certain exceptional bacteria like Rhodopseudomonas. It will uses the vanadium in their vanadium as a cofactor for the nitrogenase enzyme activity. Hence the correct answer is D. Slowly. See, there is a great saying in the English. Experience is the comb after hair was gone. Okay, experience is the comb after hair was gone. So when there is a comb, we don't know the value of a hair. So once you put the option in the exam, we never take it back. So always be slowly, analyze and think it. Okay. Question number 227. Yeah, I will write here options here itself. Option C is the B and D. Option D will be the, what is that? A and D. Take your time, no problem. I have explained you every single step in this. Eliminate it. Elimination helps you a lot. Actually, this is a kind of tough question only. A tough question in the sense options are tough. That's all. Eliminate. If you eliminate one option, you can remove the two options easily. Easily you can. You know it also. Option A. How? How option A? I'm not getting. How option B? See, friends, you only look into the options. See, I am not saying that the question is easy question. I am saying that question is difficult question only. But you look into the point. The metal associated with the enzyme nitrate reductase is magnesium. No, there is no role of magnesium here actually. Right? Obviously, option C is wrong option. If you comes to know that the option C is a wrong option, you can eliminate option A and option B also. Then there are only two options are left out. Then look into the option A. Nitrogen is normally taken by the plant in their fully oxidized form, but needs to be reduced before incorporation into the organic molecule. Yes or no? When we take the nitrogen, we can't take it directly, right? We have to convert into NH3, NH4, NO2, NO3. Is it or not? So that's a simple point here. So what will be the answer here? Option A is right. So if you comes to know that option A is right, then obviously you can choose the answer. Conversion of oxidized nitrogen into the reduced nitrogen need the energy in the form of NADP plus. No, not at all. They don't use the energy. They use the energy for ATP. Correct, na? Oxidized nitrogen means what, friends? N2. Reduced forms are what? These are all the forms of reduced forms. Then obviously we require ATP. Option B is wrong. Nitrite reduction takes place in the cytoplasm. Why? Why the nitrite reduction takes place in the cytoplasm? Because the nitrate reductase enzyme is present in the cytoplasm. Where the nitrite reduction occur in the chloroplast matrix. Why? Because nitrite reductase enzyme is present in the chloroplast matrix. Hence the correct answer will be D. Such a simple question it is. Option D is the answer. I hope everyone understand correctly without confusions. Slowly, slowly, slowly. Question number 233. You know it. Yeah, what will be the answer? Question number 233. Sri Vidya, what's the answer? A. A, is it? What's the question? What's the question? 230. The following factors is known to be involved in postponing the program cell and cell run enroll under it is completed. Involved in postponing the program cell death. That's a good question, right? Until enders for mobilization is completed. No. See, what is the answer? Anyone given? Varalakshmi, can you tell me why B? Why B? See, the question is really a very good question. I want you to understand what exactly I'm talking. You all people has chosen the answer APPSC, gibberellic acid. I know why. Because your logic is that gibberellic acid is the one which can diffuse into the allelone layer. But that is not the question they asked. They are asking a question, which hormone is involved? Listen my question correctly. Which hormone is involved in postponing the cell death that is occurring in the allelone layer? So my friends, if the allelone layer is not died, listen, see, imagine I am a allelone layer. Gibberellic acid has to diffuse inside my body, inside me. So if the gibberellic acid wants to enter inside, I am a allelone layer. First, I have to survive. I should not undergo the apoptosis. So who is going to protect me from the apoptosis? APPSC acid is going to protect. So the question is not which is involved or which is diffusing into the allelone layer. The question says that which is involved in postponing the programmed cell death in the serial allelone layer. Hence the correct answer will be APPSC acid. If they asked you which is involved in the seed germination, which is preventing the seed dormancy, which is diffusing into the allelone layer, then your answer would be correct. But the question is not that, which is involved in postponing. Don't get confused with that particular point. This is not required because brassinosteroids has to be studied. Okay. Once we discuss the brassinosteroid, we will come back to the question. Don't be hurry, take your time. 242. I want everyone to answer this question. Take your time. So fast, yaar. June. Okay. Sorry. I want you to give me 1 minute of time. Okay, anyone else? Anyone else? Okay, now listen to me carefully. You know now friends, C3 plant specialty is what? You don't need to think also, right? C3 plant, what did I told you? And what is the answer you people has given? Answer you people has given as the D, A2Q. Good, very good. A2Q, very good. C4 plant, what we studied, I told you multiple times. The moment I said that C4 plant, I told you, they will use the high temperature and they will live in the high intensity. Very, very good. Hence, correct answer will be the D. Is it? B1P CQ2. Absolutely true. Great, 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 great. 245. So much fast. So what are those? Node A, node B, node C are the all the common node genes. I hope you all people remember. Give me the example of the host specified node genes, everyone. Give me the example of the host specified node genes. Anyone remember host specified node genes, which are P, Q, H, E, F, L. P, Q, H, E, F, L are this. Okay. Okay, anyone try to answer the first committed precursor in the biosynthesis of a chlorophyll molecule? Anyone know this? The first committed precursor. You know, if you don't know the answer for this, that's okay, fine. I want you to remember. The answer will be the aminolevulinic acid. I want you to remember this. Committed precursor in the... See, we all people know that what does the chlorophyll looks like? Chlorophyll look like a porphyrin and phytol chains, correct na? But the point is here, the aminolevulinic acid will be the correct answer. Okay, that is the precursor. So question number 251. Characteristic of, and it is converted into a response regulator of a plant two component system. What will be the answer? Obviously, his glutamic acid. How does glutamic acid, Raj? So the answer will be what? Histidine. Obviously, histidine. Don't get confused with that. Absolutely true. Okay, I hope the session is little useful to all of you. So once again, once the cell biology is molecular biology is completed, once again we will do the session on the molecular biology as well. Okay, is it clear, everyone, so far? Yeah, most of the people have done a very good given a good response, but keep practicing the questions. And trust me, beyond this level, there is no point of asking the question in your JL. Not at all difficult question. I'm giving you 1000% assurance, not 100%, not more than this is only too much level. Okay, you will get a very simple questions only. Okay, that's perfect. I appreciate all your response, and we will meet sharp 4:30 a.m. in the tomorrow. So please attend tomorrow. Tomorrow I'll finish the eukaryotic transcription for all of you. Okay, perfect then. Thank you so much. Good night to all of you. Bye-bye.